Good morning, good afternoon, good evening. I'm Holly Janes. I'm looking forward to presenting today some considerations around clinical trial designs, efficacy trial designs for evaluating COVID-19 vaccine effects on transmission. I hope this is complementary with the previous talk, which is focused on observational studies, and I'll really be presenting and discussing and motivating here the ideal design of a clinical trial to answer the transmission question. I have no disclosures other than NIH funding for this research. So the driving question that motivates this research is, of course, the landmark findings in recent months that a number of COVID-19 vaccines that have been developed are highly effective at preventing symptomatic disease, with an effect seen across multiple vaccine platforms, and generally speaking, consistency of the magnitude of the effect across various demographic and at-risk subgroups of the population, but some evidence of diminished efficacy for some of these vaccines, in particular against some emerging variants of concern, with this being a high priority topic of current research. Especially given that much of the phase 3 efficacy data on these vaccines was generated in a period of time before the emergence of these new variants. But the question I want to focus on is around effects of COVID-19 vaccines on in acquisition of infection, nasal carriage, and secondary transmission, which, although there are emerging lines of evidence towards speaking towards these effects, these effects are not fully established, in particular in the context of these emerging variants. And there are multiple critical policy questions that, of course, depend on knowing whether and to what degree these vaccines prevent transmission to others. So these driving policy questions include questions such as, for individuals, if I get the vaccine, do I still need to wear a mask when around unvaccinated people or in public? Will my getting vaccinated benefit my family and community? For settings where there is vaccine scarcity, knowing about vaccine effects on transmission helps decision-making around prioritization of vaccines, in particular, the extent to which they're prioritized for subpopulations who are at lower risk of severe disease. And of course, for guiding institutional policies around both vaccination and masking for students, staff, clientele. And arguably these policy questions are even more critical as communities around the world reopen, and in the context in some communities of laws against vaccine mandates. So, of course, understanding transmission of the virus requires first understanding acquisition of infection, where acquisition of infection requires or follows an individual's exposure to the virus. That individual then, if they become infected, can be viewed as either at a given point in time being infectious or non-infectious, and if infectious, has the potential to expose their close contacts and transmit secondary infection. So the first question and most basic question around blocking or preventing transmission is asking, to what extent do vaccines block infection, which would, of course, break this transmission chain. But vaccines may also, even if not preventing fully acquisition of infection, may still have a benefit for transmission in the sense that they may reduce infectiousness for individuals who become infected despite having been vaccinated, and thereby reduce secondary transmission. So I'll be discussing here today research designs in terms of understanding vaccines' effects on both infection and infectiousness, as both are critical to understanding effects on secondary transmission. Okay, so I'm going to summarize here the two types of study designs that are providing the most evidence, preliminary evidence around vaccine effects against secondary transmission. Acquisition and secondary transmission. This is not an exhaustive list of the study designs, but really the leading lines of evidence. And really, in talking through this, highlight the limitations of these lines of evidence by way of motivating clinical trial designs. So the first line of evidence is the phase 3 trials that were designed primarily to evaluate vaccine effects on prevention of symptomatic disease. These studies, for that reason, performed primarily symptom-prompted PCR tests. They did overlay, in many instances, periodic serology, and in some instances, single or a couple of discrete cross-sectional PCR tests. But really were not designed to capture all infections, including asymptomatic infections, and in particular short duration infections or infections that did not lead to seroconversion. In terms of measuring infectiousness, they, in many instances, did capture some amount of viral load information on breakthrough infections in these studies, but it was typically done at only a few discrete time points and only after symptom onset in symptomatic cases. And there was, generally speaking, no test enrollment or testing of contacts to definitively evaluate secondary transmission. In terms of observational cohort studies, there are many of these studies, and we've heard about some of them, I believe, in the workshop preceding this talk. And these primarily, or generally speaking, consist of identifying cohorts of individuals, either prospectively or retrospectively, who have been surveilled for infection, and then ascertaining vaccination status for those individuals, and comparing infection rates and in some instances viral load in breakthrough infections between individuals who have and have not been vaccinated. Some of these studies also incorporate pre-vaccination versus post-vaccination follow-up for vaccinated individuals. A challenge with these observational studies is that oftentimes the triggers for prompting testing are not fully understood in some settings. And even in settings where there was a protocol around testing for asymptomatic infection, oftentimes these studies are enriched for symptomatic infections and employ relatively infrequent SARS-CoV-2 testing such that they may miss or not capture short duration infections. Viral load, if available in these studies, is typically only measured at the time of diagnosis, and so leaves open the question what the full spectrum of viral load is over the course of infection for the breakthrough infections, and enrollment and testing of contacts is not typical. So the limitations of these observational studies that I'll delineate on the subsequent slides are really that individuals in these observational studies are self-selecting for testing in some instances, certainly self-selecting for receipt of vaccination and timing of vaccination, and infrequent testing has the limitation that it misses infections and is going to be biased towards detection of longer duration infections. So to put a visual to this, a key limitation of observational studies is the potential for confounding by individual level factors that influence whether or not an individual is tested, the degree of exposure to the virus, and transmission dynamics, and any imbalance in these individual level factors between the vaccinated and unvaccinated groups would bias the comparison of incidence and transmission rates between the two groups. Another limitation for studies that are leveraging information, temporal information, is potential confounding due to secular trends in incidence. As illustrated using this graphic of data for the US, one may observe, as illustrated here, a decrease in incidence following rollout of the vaccination, but has to take care to take into account secular trends in incidence in order to but not be misled to infer that this decline in incidence is attributable to the vaccine, as opposed to changes in transmission patterns in the population that have nothing to do with vaccination. And a third general source of bias for these observational studies is what has been termed length bias, which is to say that when infections are surveilled at discrete time points that are infrequent, this sort of surveillance for infection is going to be more likely to detect longer duration infections, and therefore, if the vaccine is reducing the duration of infection, or infection detection, this would tend to overestimate the vaccine effect on infection given infrequent testing. So these are sort of general challenges with observational studies. There are also some key challenges for studying transmission of this virus in particular based on observational studies. One is the clear large burden of largely silent burden of asymptomatic infection, indicating that existing surveillance systems in many populations are going to be inadequate for identifying all infections. The second challenge is strong evidence of not just transmission due to symptomatic infections, but the high transmissibility of also asymptomatic infections. As well as indicated in the graphic on the right, the fact that many individuals are infected for a relatively short duration of time, and especially the duration of time when they are during peak infectiousness period is relatively short, perhaps on the order of 1 to 2 days. And finally, the fact that not all individuals seroconvert or have long-term seroconversion to this virus, suggesting that using serology to detect all infections is going to still miss a substantial number of infections. So all of these points suggest that to detect all infections, one would require quite frequent systematic PCR testing and rapid identification and testing of contacts to definitively evaluate vaccine effects on transmission. So with this as motivation, I'll now outline what I view as the ideal transmission study, and talk about a specific study that's leveraging this study design at the end. So the ideal transmission study would be a placebo-controlled study that randomizes individuals to vaccine or placebo and tests them very frequently. There's a good argument for SARS-CoV-2 for daily nasal swabs to detect all infections over a fixed duration of time, as shown in the schematic here, for a 2-dose regimen, say over a 4-month time period. Frequent testing to detect all infections and to characterize the full course of viral load for any breakthrough infections that occur in the study. This placebo-controlled trial could be implemented as an add-on to a phase 3 trial, where a subset of individuals in the phase 3 trial receive this more intensive follow-up, or it could be implemented as a standalone randomized controlled trial. To evaluate vaccine effects on secondary transmission, one requires enrollment and testing of contacts of any individuals who become infected over the course of the study. And there are strong arguments for including 2 mechanisms for capturing all infections or maximizing the capture of infections in close contacts of study participants. One is to enroll contacts prospectively at the time of enrollment of a main study participant. And the second is to enroll and begin testing contacts at the point in time when the main study participant becomes diagnosed with infection, so-called case-ascertained close contact methodology. Regardless of how the contact is enrolled, at the point in time in which a main study participant becomes infected, that contact would then be prompted to begin performing daily nasal swabs to diagnose incident infection over a 14-day period with bloods collected at the beginning and end of a 28-day period to comprehensively identify all potential secondary transmission events in those close contacts of our study participants. And then, given the challenges in distinguishing between infection events in the close contacts that are due to community transmission or other outside transmission mechanisms versus those infections in close contacts that are actually linked to the main study participants, arguably some sort of process of winnowing down the infection events in the close contacts to those that are likely or possible secondary transmission events is required. And what's proposed here is an expert adjudication committee that would take a set of data on the infections in the main study participants and the infections in their close contacts to identify those infections in the close contacts that are likely secondary transmission events. And this would include the time course of the infections, the viral load in the main study participants and the close contacts, the serology, behavioral risk data, and importantly, viral sequence data useful for ruling out potential linked transmission events. This sort of study design can be used to evaluate the following primary objectives and key secondary objectives. So the primary objectives that would be evaluated would be vaccine efficacy against acquisition of infection based on the daily nasal swabs, and infectiousness as measured by magnitude and duration of infection in participants who become infected with SARS-CoV-2 over the course of the study, based on probably primarily a peak viral load measure, but all the other summary measures of viral load, such as the area under the viral load curve, duration of shedding, would be helpful for fully characterizing the effect of the vaccine on infectiousness. And then as a secondary objective, vaccine efficacy against secondary transmissions, looking at these adjudicated secondary transmission events in the close contacts. And a number of other key secondary objectives would be evaluable using this sort of study design, importantly, and including study of immune correlates to evaluate the extent to which immune biomarkers that are associated with protection from COVID-19 disease also apply to predicting protection from infection and infectiousness as measured by viral load and secondary transmission. There would be a number of other ways in which this sort of study design would further our understanding of SARS-CoV-2 pathogenesis and epidemiology. Importantly, in the first bullet here, characterizing the incidence of asymptomatic infection with and without seroconversion, and the full course of the time course of infection and infectiousness of these viruses. And as shown in the last bullet here, generating data on both infectiousness as measured by the viral load proxy, as well as definitive secondary transmission events, which would help to study the linkage between those two and to generate data towards validation of viral load as a surrogate of infectiousness, which would not only aid in the interpretation of this study, but other subsequent studies of vaccine effects and indeed natural infection with this virus. So to make this very plain, here's what might be hypothetical data from such a placebo-controlled randomized transmission study. If this is hypothetical data from a population with a 1% monthly SARS-CoV-2 incidence and a 15% secondary attack rate, and that over the course of the study generated 50,000 person months of follow-up in the study, equally allocated between vaccine and placebo groups. So based on these sort of data, one might observe some endpoints that look like this: a total of 275 infections split between vaccine and placebo groups, with 250 infections in the placebo group and 25 in the vaccine group, which would yield an estimated 90% reduction in acquisition of infection. If we observed the mean and standard deviation of peak viral load, as shown on this slide, we could infer that the vaccine reduced mean peak viral load by 2 logs. And if we observed 33 secondary transmission events in the placebo group versus 1 in the vaccine group, that would suggest that the vaccine had a whopping 97% reduction in secondary transmission. That's attributable firstly and foremostly to the 90% reduction in acquisition, but also to what's under the rug here is a 70% reduction in the secondary attack rate, reducing the 15% secondary attack rate to 4.5% in the vaccine group. So this is the sort of data that can be generated with this phase 3 randomized controlled trial, and would provide really clear, irrefutable evidence and results on which to base policy recommendations for vaccination. So with that as kind of a description of the ideal study design, I wanted to mention that there is there is a study underway, the Prevent COVID-19 study that I'm involved with study chairs Katie Stevenson, Audrey Pettifor, and Jasmine Marcelin, that is underway in the U.S. Right now. This study design is largely similar to the ideal study design that I described on previous slides. However, it has some new features reflecting the fact that this study was not part of the original phase 3 trials or implemented quickly after those phase 3 trials, but rather was rolled out in a period of time and in a population in which there were multiple COVID-19 vaccines that received emergency use authorization and became available to the study population. So accordingly, the nuances of this particular study design relative to the ideal design is that individuals are randomized to receive the vaccine, which in this case is the Moderna mRNA 1273 vaccine, immediately on study entry, or to receive population standard of care, which includes federal, local, state recommendations on vaccine masking and so on. And so in this standard of care group, there is uptake of vaccine over the course of the study, reflecting availability and use of vaccine in the underlying study population. And any individuals randomized to the standard of care are offered study vaccine at the end of the study. As well, this study recently was augmented to include an additional observational cohort, the so-called vaccine declined cohort shown at the bottom of this slide, which is individuals who at study entry prefer not to be vaccinated. And this cohort will augment the data from the randomized trial to further inform and provide data on infection rates and post-infection infectiousness and secondary transmission profiles absent vaccination, really supplementing the data from the phase 3 trial. So as I mentioned, this study is ongoing, enrolling 18 to 29-year-olds at over 50 sites across the US, with results expected in 2021. In summary, I have summarized the rationale, motivation, and approach to the design of a randomized placebo-controlled trial, which I've argued is the ideal design for studying the vaccine effects of a COVID-19 vaccine on transmission. And this design is motivated given the unique pathologic and epidemiologic features of SARS-CoV-2 that pose particular challenges for making definitive inference about vaccine effects on transmission from observational studies. This design is perhaps best implemented as part of a phase 3 trial or immediately after evidence of vaccine effects on COVID-19 disease. And there are key challenges with carrying out this study once vaccine is available. But I've mentioned how those have been navigated in the context of the Prevent COVID-19 study in the US. So with that as the end of my comments, I wanted to express gratitude to my colleagues, including the co-chairs of the Prevent COVID-19 study, statistical colleagues at the Fred Hutch, where I'm based, and students at the University of Washington who've contributed to this research. Thank you.